Thank you. Thank you, Roy, and good afternoon to everybody. For those of you who are expecting Edward Lanfear, I'm sorry to disappoint you, but he has been giving these presentations for a long time, and we thought we'd give him a rest today. So today I'd like to tell you a little bit about Sangamo's zinc finger DNA binding protein technology and our ZFP therapeutic programs and where we are with those. I guess I should be advancing slides here. So I'm going to include a few forward-looking statements, and so I'd refer you to our SEC filings, most notably our Form 10K, which was filed quite recently and so is comprehensive and up to date. So what are we doing at Sangamo? We're a clinical stage company focused on developing zinc finger therapeutics. This is a new class of therapeutics which is designed not to produce chronic therapies, but to produce potential curative therapies, long-lasting treatments from a single treatment, so long-lasting therapeutic benefits from a single treatment. The technology operates at the DNA level, and this enables us to do a number of things. We can both regulate genes, and we can accomplish genome editing. And we're using this in a variety of different tissues and a variety of different methods, and I'll tell you a little bit more about that in a few minutes. So we also have developed broad delivery capabilities. It's not enough to be able to do this. You've got to be able to deliver these therapeutics to the correct tissue for the correct duration. And so what we can... We're using both AAV and mRNA delivery for this. The technology is proven. It has broad commercial applications, and we've established a number of different relationships with different companies. And importantly, we own the space, so we have a dominant and broad intellectual property position. So what are zinc finger proteins, and how are we using them? These are natural proteins. So our core technology is the zinc finger DNA binding protein. These are natural proteins. They're prevalent in organisms from yeast through man. They've evolved with our complex genomes. They're quite compact. Each zinc finger protein, these sort of curly blue structures, binds to 3 to 4 base pairs of DNA. And that binding capability can be changed, can be engineered so that it binds to different 3 to 4 base pair targets. And you can string these together to bind to longer and longer targets, which gives you greater and greater specificity. So we have 2 different modalities that we've developed. There's protein, we have both gene regulation capabilities as well as genome editing capabilities. As far as gene regulation, we can either activate genes. So I should first of all mention that the DNA binding portion is provided by the zinc finger, and we add a functional domain to these zinc finger arrays to enable them to carry out specific functions. So we can add an activation domain, which allows us to turn a gene up. We can add a repression domain, a natural repression domain, which allows us to repress a gene, and this is something that we're doing in our Huntington's program. And on the genome editing side, we can attach nuclease domains, which allows us to make a cut in the DNA, a double-stranded break in the DNA, which can be resolved in a couple of different ways. We can either knock out a gene, and this is something that we're doing in our programs in HIV and also in beta-thalassemia. But we can also correct or add DNA sequences, and this can be as small as a couple of base pairs or as large as an entire gene. And I'll tell you a little bit more about programs that we have there, notably an in vivo program, our in vivo protein replacement therapy platform, which we're developing with Shire for hemophilia and for our own account in lysosomal storage diseases. And so, as I say, we have a number of collaborations. We started out several years ago in broad-based collaborations in non-core areas of the of technology, or areas in which we were not planning on forward integrating. So we have collaboration in plant agriculture with AgroSciences, and in transgenic animals and research reagents with Sigma-Aldrich. More recently, we've done very defined collaborations with other pharmaceutical companies in therapeutic targets. So we have an agreement with a collaboration with Shire for hemophilia A and B and Huntington's disease, and a collaboration with Biogen Idec for hemoglobinopathies, sickle cell and beta-thalassemia. So we have this very powerful toolbox, and we have delivery capabilities. How are we thinking about how to best apply it? What tissues? What kinds of things can we do with the technology? I should say that we're largely focused in genome editing currently, but we do have a gene regulation program. And we think of this in, two different ways. So we have both ex vivo and in vivo applications. The ex vivo applications are our most advanced. We've been working in, and in those ex vivo applications, what we're basically doing is taking cells out of the body, modifying and returning them to the patient, so autologous applications. We have applications in both stem cell and non-stem cell tissue types. And so our most advanced is in the HIV space, where we've been modifying T cells. We're in a phase 2 clinical trial with that. And we are also expanding that into a stem cell application. We have a program with Biogen Idec for beta-thalassemia, which again is a stem cell-based program. But we believe these proteins, and we've data to demonstrate that these proteins can be applied, and this technology can be applied in vivo. And we're looking at systemic applications for our hemophilia programs, and also at direct tissue applications for things like Huntington's disease. So how are we specifically applying the technology? What modes are we using, and how are we delivering these? For our HIV program, we're knocking out a gene, and we're doing this ex vivo in, first of all, T cells, as I mentioned, but more recently we have an active IND to move into stem cells. And what we're doing there is knocking out the CCR5 gene. This is a co-receptor for HIV infection. And cells that don't express CCR5 are not infectable by HIV. And so the idea is that we protect a compartment of the immune system that can go on and fight the virus. And we've seen interesting data in both viral control and reservoir control. The next two programs fall into our in vivo protein replacement platform. And what we're doing there is actually integrating a gene specifically into a particular target site in the liver. We're delivering currently these both zinc fingers and the target gene using AAV. And what we're using to drive expression of this is the albumin promoter in the liver. The albumin promoter is a very strong promoter. You make pounds of albumin every year. And what we've been able to do is to integrate either factor 8, factor 9, or in the case of our Shire programs, or in the case of our own proprietary programs, enzymes that correct for lysosomal storage disorders. The common theme through these is that this particular platform is that we're going after indications for that are currently treated using enzyme replacement therapies. And so the first two indications for our lysosomal storage disorder programs are Huntington's and Hurler's. I'll move to this lower one. This is, again, another genome editing program. We're knocking out a gene again. This is in stem cells. This is a program partnered with Biogen. What we're doing there is knocking out a transcription factor that controls the switch from fetal to adult hemoglobin production. This protein called BCL11, this gene called BCL11A. And the idea there is to maintain fetal production as in adult cells, so that those cells are protective from the disease phenotype. The final program I would like to mention is the Huntington's disease program. What we're doing there is repressing the mutant Huntington gene, and we can establish very specific repression of the Huntington gene while leaving the normal gene alone. And with that, this is where we are with each of our programs. We're in phase 2 with our HIV program. We expect to have preliminary data out of that trial by the end of this year. And we have active INDs in both our HIV stem cell program and our BCL11A beta-thalassemia program. We expect those trials to start this year. We have other trials, other programs in preclinical development, and we will be filing INDs for hemophilia B program, for our 2 lysosomal storage disease programs this calendar year, and for the other programs in this list in 2016. So at that stage.
All right, thanks, Liz. Here's your mic. All right, so you said these are natural proteins, but there's actually a pretty significant amount of engineering that goes into the zinc finger DNA binding domain. And I know the nuclease is dimerization dependent, and it's been engineered as well. So that kind of differentiates. You don't see the concerns about, like, say, germline editing that have come up with the CRISPR approaches. So what kind of differentiates here? So
what differentiates the zinc finger proteins is that these proteins are, whilst they're modular proteins, they're quite complex to engineer. And I think that one of the criticisms that's always been sort of leveled at the zinc finger platform is that it's very difficult. And, you know, I think for the average lab, they certainly don't want to be having to devote time to engineer and manpower to engineer these proteins to make them very specific. The CRISPR platform, as the guidance system there, is an RNA, and so it's based on Watson and Crick base pairing. That is very much easier to use, and so that really has exploded genome editing. And you know, we are thrilled with it, to be truthful, because finally people get what we've been sort of talking about for a number of years. Having said that, we've been doing this since, you know, for a long time. The company has been public since 2000. And we are at a stage where we can engineer these proteins to exquisite single gene specificity. It's a process. It's industrialized at this stage at Sangamo. And so for us, making exquisitely specific zinc finger proteins is not a difficult task, but certainly it's not trivial for the average lab to do. So.
Right. Okay. And then I want to touch on, I think, what was the last audience question of the panel just now, and that's non-viral delivery, I believe. So you guys have started looking, I know you're using non-viral delivery ex vivo, but also looking at in vivo mRNA delivery. And what does that bring to the program? And can you eventually see maybe the entire program being non-viral? Are there efficiency gains with that approach? What do you see there?
So certainly we've seen efficiency gains in non-viral delivery in ex vivo, and that's something that we now deliver with all our ex vivo programs. We deliver mRNA by electroporation, and that has been shown to be very efficient. And that really has expanded, certainly in the T-cell population, where prior we were using adenovirus. It's certainly expanded the number of patients that we can treat. In terms of moving to, the current programs I should mention are all going to be using AAV, the in vivo programs. But in terms of moving to an mRNA-based system, that's something that's in the research sort of stage at this point. And for something like a zinc finger nuclease is actually ideal, because to make a permanent change in the genome, you only need the zinc finger nuclease to be expressed for a short period of time. And we also see advantages in maybe expanding the pipeline to go out after knockout targets, so that you could go and repeat dose. So as you've heard in the previous panel, there's a problem with repeat dosing. You either have to use a different vector or something like that. So with an mRNA approach, you would be able to repeat dose and dose to effect. And so that's certainly something we're looking at.
That should also work with knock-in too, right? Homologous recombination, wouldn't that work
certainly
there as well?
It's something that I think we could develop for that, but I think the first set of targets would probably be that knockout type of phenotype. So.
Okay, great. Are there any ultra quick questions from the audience? Under a minute. No? Okay. And okay, just I guess back to the IP, you mentioned it in your slides, but how strong, I wasn't covering the company back when you started, but I don't recall a kind of debate around IP like there is with the CRISPRs right now. How strong?
But as I say, but going back to the days, the early days when we would go out, we would talk about this and people would say, if this is such a good idea, why isn't everybody doing it? And of course, one of the reasons was we had such a broad and dominant IP position, they couldn't. And so really and truthfully, the only people working on zinc finger proteins are either Sangamo or our collaborators at this point. So it's a very strong position. I don't think anyone would dispute it.
Okay, very good. We're out of time. Thank you, Liz.
Thank you.